Inviting me and joining and allowing me to join today. I'm really very privileged and very proud to present on today's topic, as Stacy just mentioned, the drug approval process, medications in the global pipeline for COVID-19. Let me thank right now Stacy Varghese. Stacy, thank you so much, and Rachel O'Reilly as well for inviting me to speak on this topic and for all their guidance and hard work on putting this together today. So with that, we can go to the next slide, please. So today's agenda, we're going to divide it into three parts. We'll start off with the profile of some of the key elements of the novel coronavirus. Then we'll switch over to review the process and requirements that all medications, including the ones that are being developed for COVID-19, that they have to go through in order to gain FDA approval for sale and marketing in the US. And then we'll finalize with an overview of some of the pharmacologic treatments under consideration for COVID-19. Next, please. Stacy already introduced my bio. Thanks so much, Stacy, for doing that. The only thing I'd like to add is how proud I am to be a St. John's graduate. It really was a family affair for me. My brother went to law school at St. John's, and my dad also taught years ago at St. John's a film course, in fact. Like I think I mentioned, it really, St. John's helped me so much in preparing me for my career in the pharmaceutical industry. And as Stacy mentioned, most of which were spent at Pfizer in New York City. I did want to highlight the book that my colleagues and I wrote from Pfizer. It's represented on the slide. These are our lectures that we used for that program that Stacy mentioned, in which we're teaching pharmaceutical medicine to medical school students at NYU, School of Medicine, and Weill Cornell. Next slide, please. So I want to start off with this quote from Pierre-Paul Émile Roux, a French bacteriologist and developer of the first effective treatment for diphtheria. Science appears calm and triumphant when it's completed, but science in the process of being done is only contradiction and torment, hope, and disappointment. I think if you've worked in the sciences, as I'm sure many of you have on this call, you understand the trials and tribulations that we all go through when it comes to making our career in the sciences. And I think it's also very clear that the COVID-19 pandemic has taught all of humankind that science and scientific drug discovery are what the key battlegrounds are going to involve, where the outbreak will be won. And some of the hard-word lessons we are all learning and being taught along the way. Next slide, please. So let's start off with what are the coronaviruses and what is the novel coronavirus in particular. Interesting, coronaviruses are commonly circulating among all humans. There are 7 different strains that infect people, including MERS, which was the Middle East respiratory syndrome that occurred in 2012, and also that was a coronavirus, and also SARS, severe acute respiratory syndrome, that occurred back in 2003. There's another interesting coronavirus, and that's the cold virus called HKU1. And that is, in fact, as I said, a coronavirus that causes the common cold. But it's SARS-CoV-2 that's the novel coronavirus that's causing the pandemic of COVID-19. You can see from the depiction on the slide that coronaviruses are spherical in shape. They have those spike proteins that you can see that protrude from the surface, and that's what gives it the corona or crown-like structure that we can see here. Next slide, please. The coronavirus genome is important. I don't want to belabor the point, but in 1977, there were two very famous biologists, Jean and Sir Peter Medawar, and they wrote that a virus is simply bad news wrapped up in protein. And in fact, in January of 2020, as we know, scientists indeed uncovered a very bad piece of this virus, a very bad piece of bad news, namely the genome of this virus. And again, this is the novel coronavirus that causes COVID-19. Now, interestingly, this sample, the very first sample, came from a 41-year-old man who worked at a seafood market in Wuhan, China. And that's where the first cluster of cases began to appear. Scientists think that the person may have been infected from a bat, which was carrying the virus leading to COVID-19. Now, the coronavirus genome, the genetic sequence, has been uncovered, and it's determined to consist of a chain of proteins which ensures one thing and one thing only. And that's all this virus tries to do. And that's that it tries to replicate. In RNA molecules like this one, there are these letters that you can see depicted on the upper left-hand side of this slide. A stands for adenine, C for cytosine, G for guanine, and U for uracil. And what these are different chemicals. What the virus does is that it hijacks the cells. It goes in, hijacks host cells in humans, and then has the cells that it hijacks do exactly what the virus tells it to do. So in other words, the virus doesn't live by itself. In fact, it dies when it's not within a host. Once it gets into the host, it uses its genetic sequence to tell the cells to do exactly what the virus tells it to do. Now, the sequence of these chemicals actually recruit machinery within the infected cell to read the RNA letters, those letters that are up on the left-hand side, and that translates into what are known as coronavirus proteins. These are the viral proteins that make us sick. These proteins allow the virus to bind to our cells and also to replicate. Now, a vaccine, and the reason I'm belaboring this a bit, is the vaccine vaccines that are in development recognize this virus as an antigen or a foreign substance, and they incite or induce an immune response, which essentially inactivates the virus. That's what vaccines do. And importantly, because the scientists have uncovered and identified these genes, they know what they are. Vaccine development has actually been able to accelerate and certainly been facilitated. Next slide. A quick slide on the history of pandemics and the death toll that it's taken on humankind. The scientists for years have predicting that a coronavirus pandemic would not only occur, but was in fact absolutely inevitable. Why? Well, in 2007, there was a publication, and I quote, "The presence of a large reservoir of SARS-CoV-2-like viruses," that's the one that's causing COVID-19, "in horseshoe bats, together with the culture of eating exotic mammals in southern China, is a ticking time bomb." Now, on the left of the slide is a representation of the history of our most deadly pandemics, and the death toll from these pandemics is pictured on the right of this slide. You can see that at the very top is the black bubonic plague, the Black Death. It originated in rats, and it spread to humans via infected fleas. It occurred from 1347 to 1351, and amazingly, it wiped out between 30 and 50% of the entire European population. It took more than 200 years for the population to recover. The fact is that as human beings spread throughout the world, so have infectious diseases. And as our population grow and we move into wilder areas, what it's doing is it's bringing us into more frequent contact with animals that we never would normally have contact with. And viruses jump. They jump from animals to humans, and they do this in the same way that they pass between humans through close contact, through bodily fluids, etc. So today, we are really seeing outbreaks that are just about nearly constant. Now, as you can see on the slide where COVID-19 fits into the scheme of things, that's highlighted by the red arrows. Next slide, please. So as of June 2nd, 2020, there were over 6 million cases and almost 378,000 deaths worldwide from COVID-19. But I just checked again just the other day on July 7th, and there were almost 12 million cases, up from 6 million, and 544,000 deaths worldwide. So what that means is that in a little more than 1 month, the case numbers have doubled and deaths have increased by approximately 44%. Now, the emergence of these viruses, these coronaviruses into the human population has occurred, as I briefly mentioned, by spillover from bat reservoir hosts into intermediate hosts. So they jump from bats and then they can jump into civets and other small carnivores, which are sold in wet animal markets globally. Bats are known to be the host of more than 30 different coronaviruses. Bats are the most probable original reservoir for COVID-19, and we think that the spillover didn't occur directly into humans, but jumped pangolins, which are essentially anteaters. They, in fact, could be intermediate hosts. And there are over 15 million bats in the world. They're some of the largest mammals known in the world. So you can see that the opportunity for coronaviruses to spread has always been there. Now, as the COVID-19 outbreak has progressed, person-to-person transmission has been occurring when people are in close contact. And in fact, that is the main mode of transmission. The spread is occurring easily, sustainably, between people through respiratory droplets when a person coughs, sneezes, or even talks. And based on the latest information from WHO, the World Health Organization, ominously, spread may also be occurring through aerosolized droplets that remain in the air for minutes, even after people walk away from where they were just talking or coughing. Now, some people without symptoms may still be able to spread this virus. We're still learning about the spread of this virus and the severity that it causes. And it may also be possible to get infected, although it seems a little bit less so, by touching the surface or object that has the virus on it, and then by touching your mouth, nose, or eyes. So we can go into the next slide, please. So a virus has to hijack a human cell in order to cause damage. And this is a picture of how the virus enters into the human cells. COVID-19 virus uses the angiotensin converting enzyme ACE2 protein for cell entry. Now, these receptors are everywhere in our bodies, throughout the body. They are in the oral and nasal mucosa. They are in our nasopharynx. They are on our heart. They are in our lungs. They line our stomach, and they line our small intestine. And that's just a few of the organs. They really are ubiquitous throughout the body. Now, it's the virus spike protein which binds to the ACE2 protein, and that's how it gets entry into the cell. Now, I've mentioned ACE2. For those of you with a science background, or perhaps you just heard, using this door It's the same door that is important for people in regulating blood pressure. And in fact, ACE2 inhibitors for controlling high blood pressure is commonly used. When you are taking an ACE inhibitor, you actually upregulate the ACE2 receptors. And that means that people with high blood pressure have more of these receptors. More receptors means more viral binding, and that can mean more disease. Now, this may explain why some people with high blood pressure are being put more at risk for severe disease. Once the passage of the virus occurs through the membrane and the virus actually gets into the cells, there's an enzyme which actually activates it, and viral RNA, that's really the genetic material, is released into the cells where copies of the viral genome are now made. At the same time, these proteins are being synthesized in other parts of the cell. Once the virus is completely assembled, it's released and it begins to invade the body in its entirety. Viral shedding leads to bodily infection. It's what makes us sick. And indirect data suggests that infected individuals are more likely to be infectious early rather in the latter stages of the disease. And by being infectious early, often they don't have symptoms. And that's why asymptomatic spread is occurring so rapidly. Next slide, please. So as the novel coronavirus enters cells, it goes in through the doorway of the ACE2 receptor, and then it attacks the pulmonary surfactant cells, which help air get deep into the lungs. So it kind of hitches a ride, and that's how it gets deeply into the lungs and immediately starts to make people sick. When people get sick, begins to create a powerful inflammatory storm. And these chemicals that are released are called cytokines. Cytokines actually activate immune cells, which in turn induce more release of these inflammatory cytokines. So really, it's our own immune system which creates millions of these immunocells, which invade these infected lung tissues, and it causes massive amounts of damage. So it's caused by our own body, in fact, in trying to clean out the virus within infected cells. And to make matters worse, some individuals are genetically prone to have an overreaction of their body's own immune system, and that makes the cytokine storm or cascade of immunostimulants, if you will, even worse. This hyperresponsiveness can trigger hyperinflammation in every single body part as well. And this is what can seriously make people ill and even kill patients. And it may explain why some people have a severe reaction to COVID-19 while others are really quite unaffected or have only mild symptoms. It also gives a rationale for why younger people are being less affected by the virus, as their immune systems are less developed than older people. This is also providing a plausible explanation for comorbidities as the most common risk factor for severity of illness and also mortality. Not only does this lead to an explanation of why people with comorbidities are getting sicker, but it also provides a pretty neat insight in how to pharmacologically develop medicines to treat this illness. So let's go to the next slide and change gears a bit. Let's talk a little bit about the process of drug development, which really all of the COVID-19 developers have to go through as they develop their medicines. So this is a snapshot of this process, and it's really divided into three parts. The first is drug discovery, which often involves serendipity. That was the case with penicillin so many years ago. But it also involves drug libraries, which are obviously all connected through global electronic internets, et cetera. And it's allowing scientists to find molecules that they're screening to find those substances that have the best desirable therapeutics. And they're conducting basic clinical pharmacology to develop these lead compounds. The idea is to link those compounds to a target and then test them in preclinical or animal models. Now, animal tests, again, on the left-hand side of the slide, consist of classic pharmacology and toxicology, where you're looking at pharmacodynamics, what the drug is doing to the body, and pharmacokinetics, how the body absorbs, distributes, metabolizes, and excretes the drug. Once all the animal studies are completed, you go into phase 1 in human clinical trials. That's relatively small numbers in healthy normal volunteers, really a safety study. Phase 2, which essentially looks at a couple of hundred, but it's also looked at dose range finding. And then phase 3 are the pivotal studies. Phase 3 is where FDA will grant you approval. Those phase 3 studies are the key to getting COVID-19 treatments available and on the market. Now, on the right-hand side of the slide, you can see the attrition rate. For every 5 to 10,000 chemical compounds that are screened, literally only one will gain approval by the FDA. So everyone talks about new product development, risky, expensive, and really the proverbial needle in a haystack, all at a cost of an average of $2.6 billion from start to finish to develop a single new molecular entity to treat COVID-19. Next slide. The investigational process that manufacturers have to go through is tightly regulated. Why? That's because of what happened way back in 1937. Often the regulations that FDA has promulgated over the years are based on tragedies, and this is a perfect example. In 1937, there was a drug company called Massengill, and it was developing an elixir of sulfa to market to mostly young children. But they'd never tested the elixir, which they solubilized with diethylene glycol. It was the best solubilizer, better than water, better than any of the others. And they never tested it in people. And diethylene glycol, as you may know, is antifreeze. It's a complete poison. And before the tragedy could be averted, more than 100 people, many of them children, were dead. So FDA changed the regulations to now require that in order to develop a drug, you have to first test it in animals to make sure it's safe. It's done under all OSHA regulations to ensure proper handling and treatment of animals, as you would expect. Change the regulations, and now before you could put a drug on the market, you had to put them into and test them into animals and then go into phase 1, 2, and 3. It led to the creation of the IND application, which is on the right of this slide. This is essentially a safety application. FDA requires you to show them why your drug is safe, and then you have to wait 30 days before you can enter into your first clinical trial. Let's go to the next slide, please. So the way that this works in drug discovery, you're trying to find a target that's a receptor where the drug will bind and hopefully work. You're then going to conduct experiments in animals to see what the drug is doing to the animal itself. That's pharmacodynamics. Then you're running PK or pharmacokinetic studies, absorption, distribution, metabolism, and excretion. You're then going to look at various toxicology studies. And after you do all that and you prove that your drug is safe, submit the IND, and then into clinical trials you go. Just talk to you about those different phases. Now, in order to do clinical trials here in the US, you have to follow GCPs or good clinical practices. That absolutely requires that you are on the lookout and can ensure that the subjects that volunteer for these studies are protected. that their rights, safety, and well-being come before the science. Investigators have to assure that these volunteers are safe before they even consider the scientific experiments that they're doing. If you put your product into people, you have to manufacture it according to good manufacturing practices or GMPs. That's regulations that assure that you meet all specifications. And then you can file a marketing application. I'll tell you a little bit more about that on another slide. Next slide, please. Just an overview. This is what we just discussed. Moving left to right, you conduct basic research to find targets. You find those targets that link to disease. You show that changing the target results in changes in the disease. You optimize chemicals to find the best one that you want to bring into clinical trials. You conduct your studies in animals first, show that the drug is safe, get the drug into people through human clinical trials, and then file a new drug application, which is a pre-market approval application. I'll tell you more about that in a second. Next slide, please. Just to summarize, this is the IND application. Just focus on the right-hand side. If you can see it's on number 14. That's the contents of the application. And you can, under 14, that contains all of the actual contents. I'll just spotlight that on another slide real quick. But also I want you to go into the middle of the slide number 2, and look at that highlighted, bolded part. That's essentially the contract for the clinical investigator. And that reminds them of their obligations. It reminds them that they are obligated to ensure the rights, safety, and well-being of clinical trial subjects is upheld. It's hugely important, and it's something that all good clinical practices have to adhere to. Next slide. Just one last slide to summarize what the IND is all about. Really, just look at it on the right-hand side. It's a blow-up. All of the animal studies have to have been conducted, animal pharmacology and toxicology. You have to provide manufacturing information on the drug substance, that's the active ingredient, the drug product, that's the full product with all of the binders in it. And then a placebo, you have to provide manufacturing information from your clinical trial if you're in fact using one. And then finally, you show FDA your clinical protocol and investigator information. You submit all this information. You essentially ask FDA permission to go into human clinical trials. Next slide. And once you're done with all of that, you submit a new drug application. That's a pre-market approval application that asks permission to market your product. It takes all of the data, everything, the animal data, the human clinical trials, and you compile it, and you submit it, and you ask FDA to approve it for sale and marketing. Now, I gave you this picture on the right-hand side because it's actually a snapshot of my first job in the drug industry about 100 years ago. Back in those days, you actually submitted applications via paper. Now, clearly, this is antiquated, but I wanted to show it antiquated because now everything is submitted electronically. But I wanted to show you the amount of paper. There's probably over 200 boxes here containing in each box literally thousands of pages of data. And it was my very first job in industry to drive the van from the drug company that I was working for. And at this time, this was before Pfizer, but similar, I also did it at Pfizer as well. But I drove the van to 5600 Fisher's Lane, Rockville, Maryland. I can still remember the exact address. Dropped off all these boxes, got a receipt, and then went back home. This is the old way of doing things, but NDAs are still the application that FDA requires in order for them to grant you approval to market your drug. All the COVID manufacturers have to go through an IND and an NDA application process. Next slide. So again, convince FDA with the new drug application, your drug's safe and effective. Because no drug has a single effect and all drug has multiple effects, you still want to show that the benefits outweigh the risks. Want to make sure that the label is appropriate and that you can manufacture the drug according to all the specifications to ensure that the product does what it's supposed to do. Next slide. Last one on the IND and NDA. Snapshot of the IND. It's the result of a successful animal program. It's the vehicle by which the sponsor asks FDA to be allowed to advance to clinical trials, and it's considered all about safety. The NDA is the vehicle that FDA will use to approve a new drug for sale and marketing. It compiles all of the data, and at the end of the day, it's considered a safety and efficacy application. Next slide. Who reviews it at FDA? Chemists, physicians, clinical pharmacologists and toxicologists who look at the preclinical data, and importantly, statisticians. It's not that you create data that says this is clinically relevant. You have to prove it through statistics. You have to show appropriate p-values. You have to show appropriate odds ratios. It's the statistician who works closely with the medical folks to ensure that the data has statistical relevance. That's how the NDA gets approved. So finally, safety and efficacy for the proposed use, benefits outweigh the risks, proposed labeling, and that you can manufacture it according to specifications. Last thing, when you work in this industry and you work in regulatory, which I did at the beginning of my career, you always liaise with the project manager at FDA. That's the primary drug industry contact. You can't reach out directly to individual reviewers. That's considered inappropriate. Always have to work through the project manager. Next slide. So switch to the last part of our presentation. This is how the COVID-19 drugs are being developed. And really what they're doing is they're taking advantage of the basic biology of the coronavirus, and they're highlighting three essential ways to attack it. They want to be able to develop drugs that inhibit the entry of the virus into the host cell that we talked about before. So if you can inhibit that spike protein and prevent it from binding to the ACE2 receptor, you're in good shape. If you can inhibit enzymes that disrupt the replication process, then the virus can no longer replicate and eventually dies. And then importantly, you want to inhibit that inflammatory response. In some individuals, genetically inclined, they're getting this hyper responsiveness. And in fact, it's not the virus that's eventually creating morbidity and unfortunately mortality. It's actually the excessive inflammatory response. It's creating damage to those areas where the virus has already been at, caused some damage, but now this excessive inflammatory response is causing even more damage. And that is another way to reverse the harmful effects of the virus. Next slide. So FDA, back in May, issued something very unusual. They issued an emergency use authorization for the first drug that was found to probably be effective for the novel coronavirus. Now, this is fairly unprecedented. FDA does an emergency use authorization when they can justify the use of an unapproved drug, but there's an emergency health crisis, as obviously the COVID-19 pandemic is. And it's also done when a formal clinical trial may not be available. Now, it does not mean that FDA has approved the drug officially, but what it does mean is that it can become available. And they did this for Remdesivir. All right, and we'll talk more about that in a moment. Next slide, please. These are, so after they did the initial one, a little more than a month later, they added to it. So Remdesivir was the first one that they issued an EUA under, Emergency Use Authorization. And then they also added chloroquine and hydroxychloroquine. Very controversial, but at the time, that was the decision that they made. And we'll get into that in a second. Then they also administered Propovin, or they put Propovin on the list as well. And what Propovin is essentially propofol. Propofol is a sedative hypnotic that's used to sedate those unfortunate individuals that have to go on a mechanical ventilator. The tendency, as you can well imagine, anyone going on a ventilator is going to be fighting it, even when they're somewhat in a sedative hypnotic state. And this will essentially sedate them enough to allow them to intubate the patient for a mechanical ventilation. Next slide. Yeah, and the reason I crossed that out, Stacy just showed, is because, as you'll see on this slide, FDA eventually retracted hydroxychloroquine and chloroquine. And they did so because there was a study that was done and that was reported in the Lancet. But what happened was they really could not confirm the validity of the data. So they had to retract it. And when they did, FDA had no choice but to remove hydroxychloroquine and chloroquine from the EUA authorization. At the end of the day, there is simply no formal clinical data that the FDA or other healthcare practitioners can rely on that show a clear benefit of chloroquine and hydroxychloroquine. And there are known safety concerns, especially heart abnormalities associated with this drug. So for that, it was retracted and taken off of the emergency use authorization list. Next slide. There are also biological treatments that have been authorized for EUA use. People who recover from COVID-19 actually have antibodies. And these are proteins that are used to fight off the virus in their blood. And the blood that you recover from those patients, which have these antibodies in it, is called convalescent plasma. And conceptually, the idea is that if you transfuse it into people with the disease, you're going to neutralize the virus, kill it, and essentially turn people around in terms of their recovery. Convalescent plasma and hyperimmune globulin are very, very similar, and both, as you can see, have been evaluated for emergency use authorization. If this works, it's typically going to work very quickly, typically within the first 7 to 10 days of infection. And all that's needed, it still needs to be tested, but you do have to make sure that the titer of the antibodies that you're giving are in fact have enough protective antibodies in it. This holds great promise. This is hugely exciting. There have been numerous medical articles and programs on TV that really hold convalescent plasma as a great promise to the American people and to the world in terms of a very important treatment for COVID-19, for treating the COVID-19 virus. Next slide. There are other treatment options being investigated. I've got Remdesivir up here, lopinavir and ritonavir. Kaletra is currently approved. It's an enzyme inhibitor. It not showing as good a promise as was hoped for, so not sure about that one. But tocilizumab a monoclonal antibody that is approved for rheumatoid arthritis and other illnesses, is very exciting and very much on the plate for being approved. Again, for that reversal of that hyper immune response that some patients have occurred to them when they're infected with the virus. So are vasodilators. Nitric oxide and epoprostenol are two pulmonary vasodilators. When the virus attacks the lungs, they really destroy the blood vessels in the lungs. That increases mean arterial pressure. So these drugs reduce it. And what we're seeing is oxygenation is poorly done throughout the body. People are really suffering in various parts where blood vessels are not delivering oxygen. And that's because of the destruction of these pulmonary vessels. This reverses it and improves oxygenation. There are a couple of antibiotics, including the macrolide azithromycin. It has interesting pharmacological activities that makes it a candidate, but unfortunately, it's probably, so far, clinical studies have not been shown to do much in terms of treating the illness. And then we already talked about the antimalarials, which will not move forward. Next slide. I took a snapshot of clinicaltrials.gov. Clinicaltrials.gov is a registry of clinical trials run by the U.S. National Library of Medicine. And it's web-based. It provides the American people with everything and anything they want to know about clinical trials ongoing in the U.S. You can search. God forbid there's a family member that has an illness and you want to enter them into a clinical trial. Go to clinicaltrial.gov and you will find studies. What I did was I entered the following filters identified on the top there, which was essentially everything that I could think of when it comes to a clinical trial for COVID-19. And I got 118 hits that met this criteria. So this goes on for pages and pages. I think what this shows very clearly is the amount of work that the globe, not only the United States, but around the globe is doing in developing and running clinical trials for the search for safe and effective medicines in treating COVID-19. Next slide. And just a quick press release, just recently, June 1st, 2020, on the excitement around Remdesivir. It resulted in a significantly greater clinical improvement versus standard of care. The problem with this is that it's not a double-blind, placebo-controlled randomized study. It's an open-label phase 3 study. That's good. It doesn't meet the best standards of clinical trials because the gold standard as far as FDA is concerned are randomized, double-blind, placebo-controlled studies. But this is very exciting, and Remdesivir is definitely on the board for undoubtedly being approved, hopefully soon, as a treatment option for COVID-19. Next slide. But the most exciting one is Dexamethasone. Isn't it interesting? This is an old anti-inflammatory drug. It's a corticosteroid, first introduced and synthesized in 1957. 1957. Approved by FDA in 1958. They used dexamethasone to reduce that hyperinflammatory response that I've mentioned throughout this lecture. Here, they conducted a well-designed study, and it showed for the very first time, and this is the only one so far, that actually reduces mortality. It reduced mortality in very seriously ill patients on a ventilator. Very, very big deal. And super exciting and so interesting that sometimes what is old becomes new again. Next slide. Just a final word on vaccines. As of April 2020, April, May, there are probably, I just did a search, there's over 100 projects globally which are centered on the development of vaccines for COVID-19. What's good is that they're looking at a diversity of technologies, different platforms, DNA viruses, as well as messenger RNA platforms, which offer great flexibility. This diversity, as well as the unprecedented global international coordination between nations around the world, really gives me the feeling, and I'm going to just say it, that in my opinion, there is a slight possibility that, again, God willing, a vaccine could in fact be available in 2020. Now, that's not a guarantee. It's a great hope because of the tremendous ongoing work that's occurring with vaccine development. And the last slide I have, if you go to the next one, Stacy, the last one are so many unanswered questions about this disease. The threat to children, Kawasaki syndrome, a very, very can be deadly illness that causes in young children the blood vessels to become inflamed. It's just one of many open-ended questions that we still don't have answers for. There's still a lot of for us to learn about this virus. And I'm hoping that with the scientific work that's ongoing, we're going to have answers very soon, along with effective treatments and a vaccine. And with that, I conclude my presentation. So Stacy, we have time for questions. I don't know if there are any, but if there are, now's the best time. And back to you, Stacy.
Great. Thank you so much, Dr. Clair. It looks like there are no questions. So I just want to say thank you for everyone for joining us today. And a special thank you to you, Dr. Clair, for sharing your knowledge. As a reminder, all webinar attendees will be receiving a recording of this presentation shortly. Have a great rest of your day.